So I'm Carl Peters from BMG LabTech, and I'm really happy to be able to introduce you to a project that's been going on at Promega. I'm really excited to see what Terry and his group have been doing there. That will hopefully give you an insight in how you can use a real-time sensing of cell health to improve the data quality that you're able to achieve, and do this with a walk-away type of experiment where the cell health is monitored over time within a microplate reader. So my job is to introduce you to the instrument that was used for this, and it is our BMG's Clariostar. And the key to it is that it has this atmospheric control unit, or ACU. And so I'm going to tell you a little bit of details about that and how that makes this type of research possible. So the Clariostar is a multi-mode, multifunctional reader, and it can be used in all of these different modes. But the ones that we're going to focus on today are fluorescence intensity and luminescence, and the means by which you can multiplex these two different types of readings to get more information out of each experiment. So the way in which the fluorescence intensity is handled within the Clariostar is actually there are two ways. One way is through a monochromator, but this is a very different type of monochromator based on linear variable filters, which allows you for both fully adjustable bandpass up to 100 nanometer, as well as, of course, the monochromator adjusting for wavelength. Another key is the adjustable dichroic, which is important for being able to focus the appropriate light down onto the sample and then allow for the appropriate emission light to reach the PMT detector. So what sets the Clariostar apart with regards to the monochromator is because it's based on these linear variable filters, you essentially are able to create a filter within the instrumentation. And this has the important effect of allowing a lot more light through. So your sample, excuse me, your excitation and emission light, when you compare that to a traditional monochromator, the amount of light that you're able to use is greatly increased. And this is due to higher transmission efficiency and higher percent transmission through each of the stages of the wavelength selection. So in addition to the monochromator, for absolute best sensitivity, you can use filters as well. But as you can see, that the LOD is sub-picomolar, regardless of whether you're using the monochromator or the filters. And because of the modular nature of these filters, you can actually use filters for excitation and then the monochromator for emission, and vice versa, giving you higher flexibility for certain experiments, but then when necessary, having the absolute highest in transmission with the filters. So the other characteristic that is going to be talked about is luminescence, and we've made some major improvements in the dynamic range over the years. So for samples that are very bright, you have that ability to now capture that bright light. 9 log dynamic range and very high sensitivity as well. So improvements at both the high end of the spectrum and at the low end of the spectrum, giving you the best possible results for your luminescence assays. As I said, the Clariostar that's going to be talked about today has been equipped with an atmospheric control unit, or ACU. This enables regulation of either oxygen or CO2, and you can have a wide range of regulation there, from 0.1% up to 20% for both oxygen and CO2. This is the actual controller that fits on top of the reader, and it has a touch screen display that can be used. But for the most part, I think the people will use it with the integrated software, so that you can interface with the regulation of gas within the control software. And all of this information is integrated and collected and can be captured and analyzed in MARS, which is our data analysis software. So one of the more recent advances for the Clariostar with ACU is this new gas ramping function, which enables you to rapidly decrease or increase the levels of, in this case, oxygen within the system. And this has, this can be done in different steps by programming the instrument to say when you reach a certain level to hold for 5 minutes or so on. This also allows longer kinetic runs to be performed, where this ramping can be repeated over time in cycles. This type of application has potential for investigating things like ischemia reperfusion, where you can look at heart cells, say, where the oxygen tension rapidly drops, but the reperfusion, when things return to more normal, can be looked at. And so all the different stages of a disease model can be looked at within the instrumentation, which we think is really exciting. So that's the nuts and bolts of the instrumentation. Terry will now take you through how this can be applied with the assays from Promega to really make a system that's going to provide you with the best data possible.
What I'm going to talk about is what we term real-time assays. And the assays that I'll show you have been done in the Clariostar instrument to enable us to collect data repeatedly from the same sample, for periods of days. So what do I mean when I say real time? It simply enables us to repeat recording of what we call kinetic data over time, which hopefully will be obvious when I start to show you some of the data. Another important aspect of real-time assays is the reagent itself doesn't kill the cells. That gives you a lot of flexibility for multiplexing on that same sample using other assay chemistries. And that is in contrast to what we call endpoint assays. Typically, those assays have a lytic reagent that kills the cells, and it limits multiplexing. In some cases, you can still multiplex, but in many cases, it interferes with the subsequent assay chemistry that you might use. So there's a whole variety of ways to measure live, dead, or in this case, apoptotic cells. And what I'll show you today are some examples of methods to measure each one of those parameters of cell health. Before I show you the real-time assays, I want to show you the contrast of using endpoint assays, explain very briefly how some of the endpoint assays work, and some of the advantages and disadvantages of using them versus using real-time assays. So starting with assays to measure cell viability, or the number of viable cells per well, historically those assays have been done using tetrazolium compounds. There's MTT, MTS, there's a wide number of tetrazolium compounds that can be used. Cells will either directly or indirectly reduce those into a formazan that gives you a color. A similar approach is used with rizazirin. It's just a redox chemical that cells convert from some substrate to either a colored, or for the case of rizazirin, it's a fluorescent product. Okay? The disadvantage of those types of assays is it requires an incubation step, typically 1 to 4 hours, to run the assay. And then after the assay is done, you simply record either the color or the fluorescence. And in some cases, you have to kill the cells. For instance, for the MTT assay, you have to solubilize that formazan precipitate into a color that you measure, and that will kill the cells. The ATP assay is another example of an endpoint viability assay. It works a little bit differently in that what you're measuring is the amount of ATP per sample. And the general consensus, or the general idea, is as soon as cells die, they lose the ability to synthesize ATP, and the ATP that's there is removed by endogenous ATPases. So to run an ATP assay, you simply add a reagent. Our reagent's called Cell Titer Glow. It contains a strong detergent to lyse the cells. It immediately lyses the cells and releases ATP into the culture medium. So there's no 1 to 4 hour incubation step with the reagent before you get a signal. The other components of the reagent are luciferin and luciferase to generate a luminescent signal. So this type of an assay will give you a snapshot in time of what has happened in that culture well. Like, in other words, how much ATP is there. When you add the reagent, you immediately kill the cells.
Is the amount of ATP correlatable to the cell type?
Yes, the amount of ATP, typically there's a fairly standard amount of ATP per cell. However, under different metabolic conditions, you will have either increased or decreased amounts of ATP. So you need to control for that. As an example, when you activate resting lymphocytes, you'll get more ATP per cell. Another example is as cells grow in culture, attachment-dependent cells, when they start to approach confluency, typically the size will be less, there'll be less cytoplasm per cell, and there'll be less ATP per cell. Their metabolism slows down as they reach contact inhibition.
But that doesn't necessarily tell you about viability.
In general, what you do is simply correlate the number of cells to ATP. For most culture conditions of growing cells, it's a fairly linear relationship between cell number and the amount of ATP. Which, by the way, for high-throughput screening, this ATP assay is by far the most widely used method of measuring viability. The reason for that is it's quick, and it's by far the most sensitive method of estimating the number of viable cells. Okay, this is an example of a characterization experiment you would want to do for an endpoint assay. And what's shown here is HepG2 cells treated with tamoxifen for various times, doing a dose response of toxin, okay? So what you can see at time 0, there's essentially no change in ATP over these ranges of concentration of toxin. But even at the lower time points, at the highest concentrations of toxin, you're already killing all of those cells. All the ATP is gone, okay? Something else you can see here is the longer the time point, the lower the IC50 value. So that can be important if you're reporting IC50 values and you're trying to match something in the literature, just change incubation time. So that's kind of a joke, but the important part I want to convey is if you're reporting IC50 values, you have to define the culture conditions that lead to that biology. Okay, now to do this experiment, and this is the important contrasting point I want to make about endpoint assays, it takes 6 assay plates. It takes an assay plate for each one of these times. Okay? So that's in contrast to the real-time assays. So we have a real-time assay method of measuring viable cells. It's also a luminescent output assay, and the way that it works is you provide as a substrate, or I'm sorry, as a reagent, you provide a pro-substrate to the cells in culture, as well as a recombinant luciferase. So the protein luciferase is added as a reagent directly to cells in culture. Now this pro-substrate is not a substrate for luciferase until viable cells convert it into a substrate. So as long as you have a viable population of cells present, you'll generate the substrate and generate a luminescent signal. This conversion step is similar to the reduction of a tetrazolium to a formazan. It's based on NADH reduction of that pro-substrate into the substrate to make luciferase. And same general concept, dead cells cannot do that conversion, so you only get a signal from the live cells. This is an example of data coming out of the Clariostar instrument. And the way this was set up is cells were seeded into wells. And in fact, the reagent was actually added to the suspension of cells even before they were seeded into the wells. So that will actually save you a step when you're running an assay. And you simply start recording luminescence here. In this case, it's over a 3-day period. So you can see the negative control, or the lowest concentrations of toxin, generally give you a relationship of increased luminescent signal over time. That's because those cells are still growing and dividing. There's more and more cells there. But with the samples treated with toxin, you see you start to kill those cells. Depending on the toxin, the cell type, some of these are very drastic. Sometimes the toxin levels or the viability goes down immediately. But what this does also is it enables you to calculate an IC50 value at any time. So you can get some indication of the potency of that toxin. You know, as you probably know, toxins, the toxicity of a compound is generally the combination of dose and the duration of exposure. That's why the IC50 value went down over time with that ATP assay example. Okay, this is an example of how that viability assay can be useful if you want to multiplex. This graph simply shows a comparison of two different methods of extracting RNA. There was an automated method and a manual method that was used. But the important point here is that these samples, and the samples, by the way, were just microtissues or spheroids of different sizes. And what's shown here is that whether or not that real-time glow viability assay reagent was present or absent, there's no difference in the yield of RNA. In other words, it doesn't affect the biology of the cells that much. Okay, that's important because you still have a live population of cells left to do something else with. And for toxicity experiments, typically there will be people screening a variety of different types of toxins, and when you first start to see a cytotoxic event, like on the Clereostar recording, what that tells you is that some of the cells in that population are dying. Because even if you have a clonal population of cells, there's heterogeneity in that population. So if some of the cells are dying, what you can do at that point is go into that well and interrogate the sample with another assay. In this case, people were interested in interrogating RNA and doing RT-PCR to see which stress response pathways are being turned on by those individual toxins. Okay, one of the advantages, of course, of recording in real time, and this is simply another example of a 3-day experiment, is that, remember that ATP experiment I showed you, took 6 plates. You can do essentially the same thing with 1 plate with the real-time recording method. And again, calculating IC50 values at any time you want. So that's one of the advantages of recording in real time. It's more efficient, especially during assay development. I put this in here as a plug. This is, there's information about the real-time assays in this chapter that's in the Assay Guidance Manual that's on the NIH website. This is a completely open access resource. You can have access to it, download it, use it for teaching, whatever. Okay, I'm going to switch gears a little bit and now talk about real-time methods to measure dead cells. Question? Excuse me?
Can you stain, fix and stain the cells?
Yes. Fluorophores, other antibody staining, yes.
Is the reagent just added with the medium?
Correct. For the real-time glow assay, again, it's simply a combination of a recombinant luciferase and the pro-substrate that's added directly to the culture medium. The luciferase, by the way, is one of the luciferases derived from shrimp. So it's a different chemistry than the type of luminescence that is measuring ATP. The ATP, or the luciferases that use ATP, are typically like beetle luciferases, whereas marine organisms typically use cilantrazine as the native substrate, but they don't use ATP.
Do you have a robust data set showing comparison between cell titer glow data and the real-time cell titer glow has been around a long time. People feel comfortable with it, and when I offer to use these sorts of live dyes, there's pushback from various program teams because they're more comfortable with cell titer glow.
That's a good question. We have had customers do fairly high throughput screens with this. I don't think we internally have a direct comparison to find out which compounds, for instance, would interfere with this type of a luminescent assay versus the Firefly luminescence. But that's a good question. I'll have to ask our tech service people because I can't remember everything that's been done. Okay, I'm going to talk a little bit about measuring dead cells in real time. There's two basic ways that people measure dead cells in vitro. They either measure the leakage of an enzyme out of cells that have lost membrane integrity, or they measure the uptake of a dye. Most commonly, for plate-based assays, it's a fluorophore that will bind DNA. Probably the most commonly used dye to measure membrane integrity is trypan blue, right? You get your cells out of the incubator, you do some staining. All that's really telling you is whether or not the cell membrane is intact. That's the most common definition of a dead cell, is based on membrane integrity. This is a similar example that I showed you for the ATP assay, except in this case, it's looking at the release of an enzyme, lactate dehydrogenase, from dead cells, or cells that have lost membrane integrity, into the culture medium, okay? Same type of a dose response of tamoxifen on HepG2 cells. At time 0, you see essentially no change in the fluorescence signal. There's no release of LDH at time 0, which is what you expect. But again, even after short incubation periods, at the highest concentration of toxin, you see a maximum amount of LDH release. But the interesting thing, and what I wanted to point out with this experiment, is look at the 24-hour data. The level of LDH measurable is much lower. And why is that? It's because these cells died at 2 hours, and they've released the LDH into the culture medium. That enzyme is not stable forever in the culture medium. It's probably gone through like 1, 2 half-lives. So that's something you have to be aware of if you're measuring LDH, is it's sort of a moving target. We have a way to get around that. So there are actually a number of DNA binding dyes that can be used. Promega's developed one called CellTox Screen, and the way that it works is you add it directly to cells in culture. It's a non-permeable dye. It doesn't enter live cells, but it does enter cells that have lost membrane integrity and stains the DNA green. Okay? The magic part about this dye is it's relatively inert to the live cell population. So it doesn't affect the biology, you simply detect dead cells. So this is again a 3-day experiment done in the Clariostar, and the way that it works is you simply record fluorescence over and over for this 3-day period. And in this case, these were programmed to record every hour. That's why these dots are so close to each other. Okay? What you can see is the vehicle control and the lowest concentration of toxin have no change in fluorescence over this 3-day period. The intermediate concentration of toxin, you see a gradual increase in fluorescence over time. What that's telling you is you're accumulating dead cells in that culture. Okay? Another important thing I want to point out is look at the highest concentration of toxin. You kill the cells right away, but that fluorescence signal remains fairly constant over that 3-day period. That's a huge advantage compared to trying to measure LDH, because LDH signal would come up and it would go right back down.
So is that a bottom read configuration? I mean, cells that may come off the plate over time, or what's the? I'm just curious.
I'm going to ask the experts to answer that question.
Well, yeah, so it certainly can be done with the bottom reading configuration. Both top and bottom reading can be done with extremely high sensitivity in the Clariostar. And for the majority of cell-based assays, that's what we initially recommend. But through experience, we've seen that either a top or bottom reading can work quite well. And that's due to the fact that there's actually a focus adjustment that is performed. And so you're detecting, well, at the start of the experiment, you're detecting where the cells are on the bottom plate. So.
Let me put the reagent question another way. Does the reagent best detect compact DNA, or will it detect the DNA as it's released from cells into the soup? These are whole well readouts. And so cells that lyse and are dead 3 days later that were killed on day 1, how are they still visible? Because here you don't have a difference.
Yeah, and I'm not sure if that's why this is lower, but there is a difference in the amount of fluorescence depending on if you kill cells right away with detergent or if you kill them and then watch them hang out for 3 days. It probably has something to do with the unwinding of the DNA or the histones wrapped up, or if it's wrapped up in a nucleosome, whatever.
Sure. No, thanks.
Okay. There is a difference in the dyes that you choose. Now, most of these dyes are somewhat similar, and if you're familiar with fluorophores, these are actually asymmetric cyanine dyes, most of them. Not the Herx, but these two are, the Celtox-Screen and the Yopro. And even if the dyes are qualified or claimed to be non-toxic, I would encourage you to test it with the cell type that you want to use. Test it for a long period of time. What is shown in this experiment is 4 different cell types exposed for 3 days to those different dyes. Some of the dyes can be toxic. Now, when someone tells you that a dye is not permeable to live cells, they probably really mean that it's just not functionally permeable. It's possible that some of it could get in and be immediately pumped out. You know, because if you add protein to cells in culture, some of that protein is going to end up inside the cell, right? So, but what we do know for this dye, the Celtox-Screen, is it functionally does not stain the live cell population. This is simply another example and a different way to present real-time data. In this case, it's actually combining cytotoxicity measured with that fluorescent dye and a decrease in viability by measuring the decrease in the luminescent signal. So you can multiplex essentially any fluorescent assay with the luminescent cell viability assay, okay? And you can, you know, look at different concentrations of toxin and over time as well.
How well do EC50 values agree between the two approaches?
That actually depends on the toxin, because sometimes you'll see a decrease in the metabolic marker prior to seeing the loss in membrane integrity.
Fluorescence and the luminescent signal absolutely not interfering overlapping with each other.
No, there's generally no interference, but keep in mind that when you collect luminescence, you don't typically use a filter set, and you're simply trying to collect all the photons you can out of that sample. With a fluorophore, there's an excitation step and collecting at an emission wavelength. So it's a little bit different.
So any fluorescence can be used in combination with your luciferase assays?
In general, yes. Okay, I'm going to now switch gears again and talk about measuring apoptosis in real time, so a method or programmed cell death. And again, I'm showing you the same type of a characterization experiment that was done by measuring caspase-3 activity with a luminescent assay. And again, the same model, toxin on top of HepG2 cells, recorded at different times. This is an endpoint assay. The caspase detection reagents contain detergent. Okay? It lyses the cells, releases the caspase enzyme, and the reagents are added to record how much enzymatic activity is there. And again, the same trend occurs. At time 0, there's no activity, but even at the short times, there's a maximum amount of caspase activity detected. Caspase can be induced in cells relatively rapidly, a lot more rapidly than people are aware of, okay? But again, the same trend is there for the 24-hour data in that you have less activity at these high concentrations of toxin at 24 hours compared to 2 or 4 hours. And again, that's because after cells die by apoptosis, they will eventually undergo secondary necrosis and lose membrane integrity in vitro. That's different than in vivo when cells are engulfed by macrophages because of the signals on the surface of the cell. But in vitro, they'll eventually just fall apart. The caspase will be released into the culture medium, and just like LDH, the caspase is not stable forever. Okay? So we've developed a new method based on annexin 5 staining that you can use to detect the occurrence or the onset of apoptosis in real time. And just briefly, I want to describe how an annexin 5 binding assay works. And it's based on the fact that normal viable cells typically have an asymmetric distribution of phosphatidylserine. The phosphatidylserine head groups are normally oriented toward the cytoplasm. During the process of apoptosis, and this is actually a caspase-mediated signaling event, some of the phosphatidylserines flip out to the outer leaflet of the cell membrane. Okay? And they actually flip out in these ordered structures that you can visualize with very high definition microscopy. So the reagent to measure apoptosis in real time is composed of annexin 5 that's linked to subunits of luciferase. Now, annexin 5 is just a protein that's known to bind to phosphatidylserine. It's just used as a tool. Historically, annexin 5 has been linked to fluorophores. And to do the assay, you would add the fluorescent annexin 5. When cells undergo apoptosis, it would bind to the surface. But then you typically have to wash away the unbound annexin fluorescence before you run the assay. In this case, we're providing two different annexin molecules, one that has a large subunit of luciferase, and the other annexin molecule has a small subunit of luciferase. And again, this is the shrimp-derived luciferase subunits that we're using. And in this situation, there's no luminescent signal. So there's no need to wash away unbound molecules. In the situation where apoptosis is occurring, the two annexins can bind in close proximity on the cell surface and reconstitute this shrimp luciferase called nanobit to generate a luminescent signal. So as cells are undergoing apoptosis, you'll get more and more luminescent signal. Now what will eventually happen is secondary necrosis, and the control that you must do is to determine whether or not membrane integrity is intact. And the reason for that is after you lose membrane integrity, some of the annexin can get inside of those dead cells and stain from the inside. So the typical control is to use a DNA binding dye. Historically, for flow cytometry, it's been annexin FITC with propydium iodide to measure membrane integrity. Okay? We're using a DNA binding dye also, which is indicated by these little dumbbell-shaped things. So it goes in and stains the nucleus green. So we can simultaneously record the onset of luminescence to indicate apoptosis and the increase in fluorescence to indicate secondary necrosis. So you can set up an assay plate, add these reagents, stick it in the Clariostar, and start recording data in real time. Depending on the type of stimulant you add, you'll have different kinetics for the onset of apoptosis. In this example, it's a relatively rapid increase in the exposure of phosphatidylserine that's detected by those two annexins binding, reconstituting luciferase, and generating this luminescent signal. In this particular case, you see there's quite a lag before we lose membrane integrity and the DNA binding dye starts to stain the cells. So you can see the fluorescence increase as the cells lose membrane integrity. So you can do this in real time and determine when to make the measurement. So that, again, is in contrast to the endpoint assay And if you saw that example where caspase had already come way down, so you can catch the wave of apoptosis using a real-time method, whereas unless you do multiple plates of samples in an endpoint assay, you're likely to miss that sort of wave of apoptosis. This is just some additional data. In this case, you can see another type of a stimulant. In this case, this is an antibody-drug conjugate. Okay, so you're seeing, in some cases, the secondary necrosis happens much closer to the apoptotic event. And depending on the cell type, depending on the stimulant, you can see different patterns of apoptosis versus secondary necrosis. Now, I have a time-lapse video to make a point. How often do you have people try to show a movie and it doesn't work? Keep your fingers crossed. Okay, this is a field of HeLa cells. Okay, this is just actually in 2 dimensions. And what you'll see on the left side are images taken with a microscope that detects luminescence. So you'll see individual cells light up as the apoptosis occurs. That's working. So you see the cells start to shrink here. This cell will die first. You know, shrinkage, one of the hallmarks of apoptosis. And then you see the luminescent signal on the left side. Okay, that happened over like a 15-hour period. The point I wanted to make is, again, that there's heterogeneity among individuals in the population of cells. So they don't all die at the same time. Okay, you will miss that with an endpoint assay. But you can see an increase in apoptosis over time if you do these recordings in real time. I have one more. In this case, this is going to be multiplexing the luminescent signal on the cell surface, and eventually you'll see the nucleus of the cell get stained with the DNA binding dye. So this is an individual cell. You'll see the outside light up first, and then as the cell undergoes secondary necrosis, you'll see the nucleus get stained with the fluorophore. We have to acknowledge Olympus, the people in Japan and Olympus gave these images to us using our reagents. Okay, we have one more way to measure apoptosis in real time, and that is by using a biosensor. And the biosensor is actually a firefly luciferase that has been engineered to contain a caspase-3 cleavage sequence. Okay? So you express this luciferase inside of cells, so you have to transfect the luciferase in. It's expressed in cells, and in the normal situation, this DEVD containing peptide holds that luciferase in an inactive conformation. When caspase cleaves that peptide, it enables the luciferase to refold and generate a luminescent signal. So you can simply record in real time the increase in luminescent signal, indicating that caspase has cleaved that peptide and generated an active luciferase. So that's basically what's being shown here, with different exposure times with the different colors that hopefully are not as colorblind as I am, with increasing concentrations of toxin. And you can see, you know, the lower concentrations or the lower exposure times, there's very little, but at the intermediate, you'll see more and more caspase activity, and you can record, you know, potency at different times.
Do you need to have a stable expression of the luciferase?
It's recommended to have a stable expression. So this is just an alternative method of measuring apoptosis in real time, but of course the disadvantage is you have to engineer the cells. But since we already had this available, I did want to include it just as another option. But it wouldn't work for instance, primary cells, or it would be very difficult to do this. Okay. Historically, annexin 5 binding has been described as an early marker of apoptosis. And that claim was made about 20 years ago when Annexin 5 technology first was made available. And at that time, all they had available to measure caspase activity were substrates that gave you a colorimetric signal. So they were linked to para-nitroanilide, okay? And the sensitivity of those old reagents is probably less than 100-fold or 100-fold poorer than the existing luminescent assays. And what we know now is that caspase is actually involved in signaling events that lead to the events like the scramblases and flipases that flip out the phosphatidylserine, and caspase activity typically can be detected earlier than Annexin 5 binding. And that's simply what's shown here by running these at different times and looking at the data. And if I can pick out the longest time here, you see is actually here, but for caspase activity. But if you look, you can't really detect much going on with annexin until 3.5 hours. But if you look at the 3.5-hour value for the caspase activity, it's already at maximum. So caspase activity typically does precede Annexin 5 detection of phosphatidylserine. That's it. So if you have questions, feel free to ask. I will also be available at the Promega booth a little later this afternoon. I have a conflict immediately after this, but I'll be around, or you can send me an email. Yes?
So what's the correlation between cell titer glow and the cell tox screen?
I'm sorry, what's the correlation?
The correlation in, if you see changes in cell titer glow, do you...
Yes. Good question. Typically, if you plot both luminescence and fluorescence, it's an X. Very often, the IC50 values are very close. There are some situations where if you're changing the metabolic marker, whether it be ATP or the real-time, or even a tetrazolium or rizazolam assay, they may not exactly match up.
But kinetic, so one might be a little bit more right-shifted, for example.
Yeah. But keep in mind, when you're using these reagents, try to remember exactly what it is that they're measuring. A lot of people talk about ATP as being a proliferation assay. It's not really a proliferation assay. If you use it over a long period of time, you'll have more cells as a result of proliferation, but it's not really measuring a proliferative event.
In the annexin luciferin reconstitution system, I forgot, did you show us long incubation times for that as well? Are those reagents stable for a few days outside of the cell?
The reagents themselves are stable, but I should mention one of the limitations of that is, well, first of all, apoptosis usually happens in a relatively short time frame. But if you leave it there for a very long time, if the cells are dead, you will lose the signal. And the reason is we stabilize the substrate by putting a protecting group on it. And it takes an active cell or a viable cell to cleave off that protective group to enable it for the luciferase substrate.
So it only shows you actively caspasing cells, not cells which had previously...
Right. It would not be useful for measuring cells that died 4 days ago.